grade 9. Hope you're doing well. Today we're going to continue the second part of activity 1, gametes, specialized cells with 23 chromosomes of chapter 9, sexual reproduction and genetic diversity. Okay, let's first start with a quick review about meiosis. What is meiosis? Meiosis, also called gamete formation. It is the division of the mother cells that are with 2 N chromosomes found in the genital organs, testicles in man, ovaries in woman, into 4 daughter cells with half the number of chromosomes, which means having N chromosomes instead of 2N. These daughter cells are gametes, sperm cells in males and ova or eggs in females. Note, all human cells possess 46 chromosomes, 2N with 2 copies of each chromosome, except for gametes that contain 23 chromosomes or N. The sperm, or the male gamete, it contains 23 chromosomes, of course, grouped into 22 autosomes plus X, one gonosome, or another type of gamete containing 22 autosomes plus Y gonosome. The ovum is the female gamete. It also contains 23 chromosomes, which are 22 autosomes plus 1 gonosome X. So the sperm cells can be of 2 types, 22 autosomes plus X or 22 plus Y, while the ovum, all ova are of 1 type, having only 22 autosomes plus 1 gonosome X. So, is meiosis similar to mitosis? Let's see. Meiosis, actually, it consists of 2 successive divisions. So, it's not similar to mitosis. Okay. Like in every division, always we have a phase that we call growth phase or interphase. And this interphase, it occurs always before mitosis and also it occurs before meiosis. So, look at the diagram, please. We have here a sperm mother cell, or it could be also an ovum mother cell. First of all, it undergoes a growth phase, which is interphase, and grows up. During this interphase, the sperm mother cell or the ovum mother cell, they double their DNA. So a duplication of DNA occurs, which means the chromosome of 1 chromatid becomes of 2 chromatids. Then the cell that is now with 2 chromatids per chromosome is ready to start meiosis. As you can see here, meiosis consists of 2 successive divisions. Meiosis 1, which is reductional division. Why? Because as you see, the cell of 2N chromosomes divides into 2 daughter cells with half the number of chromosomes, or N. And that's why this division is called reductional. Reductional means there is reduction, which means also a decrease in the number of chromosomes from 2N into 1N. Okay, so at the end of meiosis 1, we have 2 daughter cells, each with N chromosomes. Then, each of these daughter cells with N chromosomes, it undergoes meiosis 2, which is an equational division. Why it is called equational? Because as you see in the figure, every daughter cell with N chromosome divides into 2 daughter cells also with N chromosomes. So it's the same number of chromosomes. It's an equal number of chromosomes. So from 2 daughter cells with N chromosomes each, we have 4 daughter cells with N chromosomes each. And that's why meiosis 2 is considered to be equational division, because the number of chromosomes is the same. So let's explain now the process of meiosis, and to facilitate the understanding of meiosis, two pairs of chromosomes are shown in the figures below. So, as I said previously, that meiosis consists of two successive divisions, meiosis 1 and meiosis 2. Here, in the figure shown here, we can see the meiosis 1 with its four phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. Let's see what happens during prophase 1. So, this is a cell with a nuclear membrane that is disappearing progressively. The centrosome changes into asters that migrate to opposite poles. The spindle fibers appear between the two asters. The chromatin, long and thin chromosomes, they condense into short and thick chromosomes. So you see inside the nucleus, we have here 2N, 4 chromosomes. Every 2 chromosomes forms one pair, okay? And we say homologous chromosomes pair up, which means the 2 homologous chromosomes, they form together one pair. They stay close to each other. However, in mitosis, in prophase of mitosis, there is no pairing up of homologous chromosomes. The chromosomes are distributed randomly in the nucleus. So, this is for prophase 1. And note here that every chromosome, of course, has 2 chromatids. Okay. What happens in the next phase in metaphase 1? You know that when I say metaphase is characterized by the equatorial plate. Is it the case here? Yes. You can see chromosomes forming the equatorial plate. But is it like in mitosis? No, because actually here in the case of meiosis 1, the homologous chromosomes pair and line up forming the equatorial plate. So in metaphase 1, we don't have, like in mitosis, every chromosome alone on a single spindle fiber and gathering all of them in the center of the cell. No, in the case of metaphase 1 of meiosis 1, every 2 homologous chromosomes pair up and they line up in the center of the cell, forming with the other chromosomes the equatorial plate. We always have 2N chromosomes in metaphase 1. Let's move to anaphase 1. Always anaphase is characterized by polar ascension, that is migration of chromosomes to the opposite poles. Let's see here. What happens in anaphase 1 of meiosis 1? The homologous chromosomes that were in the equator of the cell, they separate and each chromosome migrates toward opposite pole. So, as you see, at each pole of the cell now, there is only N chromosome, which is 2 in our case. So, this is polar ascension. Telophase 1, 2 daughter cells, as you see, are formed, each cell containing N chromosomes, which is half the number of chromosomes in the mother cell. If you look at the chromosomes inside the nucleus, of course, it reappears, okay? And the chromosomes here, they decondense, they start becoming longer and thinner. But what is important here to say is that we have N chromosome. Each chromosome is of 2 chromatids. So if you look back at prophase 1, we had 4 chromosomes in the nucleus. while in telophase 1, we have 2 daughter cells now with 2 chromosomes in every daughter cell, which is half the number of chromosomes found in the mother cell in prophase 1. We move now to the second division, meiosis II. Okay? So, every daughter cell of meiosis I now has N chromosomes, or 2 chromosomes in our example, and this daughter cell is ready to start meiosis II. So meiosis II consists also of the 4 phases: prophase II, metaphase II, anaphase II, and telophase II. What happens in prophase II? Actually, same events as in meiosis I, which are formation of asters, disappearance of nucleus, and formation, of course, of spindle fibers between the 2 asters. What is important to us here is that the chromosomes inside the nucleus are N chromosomes. Here we have 2 chromosomes, each of 2 chromatids. What happens in metaphase II? Each daughter cell, we have here 2 daughter cells, of course, and both of them are undergoing meiosis II. So each daughter cell starts metaphase II. What happens in metaphase II? As you can see, the N chromosomes now line up, forming the equatorial plate. So each cell contains N chromosomes, and these N chromosomes are on the equator of the cell. So this is something that is similar to mitosis, because you remember that in mitosis, the chromosomes were in the center of the cell, but each one of them was alone on a single spindle fiber, just like here in meiosis II. Okay, in anaphase II, since every single chromosome is on a spindle fiber, then what is going to happen here is a splitting of the centromere. So the chromosome divides into 2 chromosomes, each of 1 chromatid, and each chromosome of 1 chromatid is going to move to one pole of the cell. So sister chromatids separate and migrate toward opposite poles. Finally, in telophase II, every cell in anaphase II is going to start cytokinesis, division of a cytoplasm, to give 2 daughter cells. So as a result, we have 4 daughter cells, each with N chromosomes, but this time the chromosome is of 1 chromatid. So if you look at the cells in telophase II and at the cells in prophase II, and you count the chromosome, you see that in prophase II we have 2 chromosomes, as well as in telophase II we have 2 chromosomes in every cell. So the number of chromosomes is the same, but the number of chromatids is not the same. In telophase II we have every chromosome of 1 chromatid. So telophase II is the end of meiosis II, and these 4 daughter cells are going later to differentiate into gametes: sperm cells in the case of men and ova in the case of women. So here we have a comparison between mitosis and meiosis. Mitosis, it occurs in all body cells. As we said, it occurs in somatic cells like skin cells, intestinal cells, pancreatic cells. However, meiosis, it occurs only in the reproductive cells, which are the mother cells of the sperm and the mother cells of the ova in the genital organs. Mitosis consists of 1 division, prophase, metaphase, anaphase, telophase, while meiosis consists of 2 divisions, meiosis I, prophase 1, metaphase 1, anaphase 1, telophase 1, and meiosis II, prophase 2, metaphase 2, anaphase 2, and telophase 2. In mitosis, there is no pairing up of homologous chromosomes, at prophase, while in meiosis, there is pairing up of homologous chromosomes at prophase 1. In anaphase of mitosis, there is a splitting of the centromere, then migration to the poles, while in meiosis, especially in meiosis 1, in anaphase 1, there is separation of homologous chromosomes. However, the splitting of the centromere occurs in anaphase 2, just like in mitosis. Mitosis produces always 2 daughter cells, while meiosis leads to the production of 4 daughter cells. The daughter cells in mitosis have the same number of chromosomes as the mother cell, 2n, and you know that always in mitosis the number of chromosomes is conserved, while in meiosis the daughter cells have half the number of chromosomes of the mother cell. Okay, as a homework, please draw and label a cell with 2n equals 3 pairs of chromosomes or 6 chromosomes at the following phases: prophase 1, anaphase 1, metaphase 2, and telophase 2. Thank you, stay safe, and see you soon.